Hey everybody, welcome to week 8 in micro. So this week we just have one PowerPoint to go through, which will be the cultures or infections of the GI tract. I can't remember exactly how I titled it, but let's pull it up here. Yes, infections of the gastrointestinal tract. So let me put this into slideshow form and we'll get started. All right, so again, your gastrointestinal tract is anything basically from the starting point of your mouth all the way down to the last point of your anus and everything in between as seen in this picture included. So lots of normal flora because we're covering a lot of different areas. So a lot of the Enterobacteriaceae group are found as normal flora within the GI tract. And if you know anything about medical terminology, you will know that entero, the E-N-T-E-R-O part, stands for intestines. So these are bacteria that are normally found within us, especially in the intestinal tract area. So that is E. coli, things like that. So we do have that normally within us, as well as looking for these things, of course, in different infections. Some different anaerobes like Bacteroides, Clostridium, Streptococcus, Bifidobacterium are all found in here, and then we also have another Grandpa's rod, Granibacterium. So all of this is normal flora in the GI tract. So with that being said, when we do consider infections in the GI tract, there are lots of different toxins at play here causing these infections. So let's go through, there's a couple of categories here of toxins that I want you to get to know. The first one here is enterotoxin. Again, enero for intestinal. So these are toxins that basically alter the metabolic activity of intestinal epithelial cells that causes them to release fluid and electrolytes out. So if you think about that, they're causing extra fluid and electrolytes to be released out. The stool or the patient's diarrhea will become very watery in consistency as a result of these enterotoxins. So right away, when you think of really watery stools, cholera might pop into your mind, and we have talked about Vibrio cholera, with that causing those rice water stools. Remember, they were extreme watery stools with little flecks of mucus that they said resembled rice, so that's why they nicknamed them rice water. So that definitely fits into an enterotoxin situation. Other ones that causes kind of watery diarrhea is Shigella dysenteriae, our enterotoxigenic E. coli, that's the one that goes with like traveler's diarrhea, Salmonella, C. diff, and Campylobacter jejuni. All some of our big bad boys here are in this category. Cytotoxin, cyto for cell. This is going to disrupt the structure of the intestinal epithelial cells and basically cause those cells to slough up, leaving the surface unprotected, and that allows the surface to get damaged. So you will see kind of blood in the stool on this one with the damage to the surface. Lots of neutrophils here as well, PMNs. So dysentery, any kind of dysentery often reflects this situation. So ones that are well known to do this is the enterohemorrhagic E. coli and Shigella. They kind of share a very similar toxin called that Shiga toxin, and that is a type of cytotoxin. And then the other one would be your C. difficile. And then neurotoxins. These are a lot of times what results with food poisoning cases. I think I've talked about this, where you can get food poisoning not because you ingested the bacteria, but because you ingested the toxin that they left behind. So if they have left the toxin behind and it's not cooked out of the meat, you can still get that food poisoning because it's still going to do the damage it needs to do. So Staph aureus, Bacillus cereus, we haven't seen that name in a long time. Remember, Bacillus cereus is a grandpa's rod, and I only asked you to know 2 diseases with that back in Micro 1, and that was food poisoning and eye infections. So here we see it showing up for the food poisoning. Clostridium perfringens, again, that does result in a type of severe food poisoning. The other disease it caused was gas gangrene. This is probably the most common that we see neurotoxins really associated with. And then botulism, Clostridium botulinum. And then other things that they can do to help cause infection, attachment, or adherence. So lots of different ones there, some parasites and viruses at play as well. Invasion, allowing them to get into deeper tissues. You see a lot of different bad boys in that list. Another couple parasites are in there as well. So I'm more worried, just for reference sake, before I keep going into the infections, about the 3 categories of toxins. that you remember those 3 categories of toxin and what bacteria cause which ones. So I'm more worried about the neurotoxins, the cytotoxin, and the enterotoxins. So remember what it means to be each one of those 3 in which organisms were in each one. So that's what I'm more focused on, more so than the invasion attachment. We mentioned those. Those are also part of how they cause infection, but I don't need you to remember those things. All right, so let's get into the actual different infections in the GI tract. So we'll start with esophagitis, esophagitis, whatever you want to say, esophagitis. So inflammation of your esophagus. Yeast is a big one here, candida, herpes virus, and cytomegalovirus. So not really bacterial in nature, more other types of organisms. Gastritis, this right away should be H. pylori, so ulcers, if you will, gastric ulcers. So H. pylori is our biggest cause of gastric ulcers. Again, we just learned H. pylori this quarter, and that was seen with the chapter with Campylobacter vibrio that week. We studied it. So remember, H. pylori is a gram-neg rod. One of its biggest testing characteristics was that it was urease positive. So that was one of the biggest things, and I talked about that you can, I mean, there was a test that we did where we would get little biopsy samples sent down from surgery, whatever they would suspect, maybe an ulcer. They'd send us a little biopsy sample, and we'd stick it right into this little urea agar, incubate it, look the next day. If it was bright pink, it was positive. It was H. pylori, because that was urease positive. If it was yellow still, it was negative. It was that simple of a test. So H. pylori, hugely known for urease activity. Proctitis is inflammation of the rectum. This is all about STDs on this one. So chlamydia, herpes, syphilis, and gonorrhea. All right, specimen collection and transports. Typically, whenever somebody comes in, the most common complaints within the GI chapter, of course, is going to be that they have diarrhea. So usually we're always giving out a stool sample, a stool container, and we're also giving out an ova and parasite container, what we call an O&P exam. They usually very commonly go together. If someone comes in complaining about diarrhea for a long time, the doctor really wants to screen for different bacterial organisms that are causing it and different parasites that might be at risk. So commonly those 2 little jars get passed out together to the patient. So it's usually a stool culture jar and then what we call an O&P, ova and parasite jar. They always have like preservatives inside of them as well. There are some transport mediums, especially if you're getting more of like a swab. There is a Carrier-Blair transport medium instead of those containers. Culture plates, blood agar, of course, Campy agar. If you're looking for, if you're screening somebody that has diarrhea for different bacteria, one of the main bacteria that causes diarrhea is Campylobacter jejuni. And remember, that likes a little bit warmer of an environment. It wants a 42-degree environment to grow. Now, you can plate that also onto an additional blood agar plate and put that in a 42-degree environment, or there are specific Campy agar plates that you can put it on. Either way, just make sure it's in that warmer environment. So yeah, definitely Campylobacter at that 42 degrees. MacConkey, I mean, most of this, what we've talked about, is gram-negative rods, if anything, out of here. If it happens to be a gram-positive, that will grow on the blood agar. HE, XLD, because we talked a lot about Salmonella and Shigella, and those are primary suspects anytime somebody has diarrhea. HE and XLD, remember, are specifically looking for Salmonella and Shigella. So those are great plates. Either one of them work to set up. And then a gram-neg broth that kind of is meant to grow enteric gram-negative organisms. Again, we're primarily looking for gram-negs here. And then, of course, if we suspect C. difficile, this has, yes, you can culture it, of course. You can set up an anaerobic plate. But typically what they do here is they run a KIT test. And so they look kind of for this, the toxin in a KIT test. It's a quick turnaround. They get a quick answer right away to know if they truly have C. diff. The other big one that they can do is just draw blood and look for the antibody-antigen reaction there kind of deal. So not always we're culturing this, but you definitely can culture it, and then in that case, it would be more of an anaerobic environment. All right, I know we didn't really discuss much of the actual diarrhea illnesses. I think you kind of get the idea of it's diarrhea, like with the majority of this, and that's where it falls under the toxins, how the consistency, Sometimes it might be bloody if they're more of a cytotoxin involved with hemorrhagic E. coli 0157. Or maybe it's the Vibrio cholera and it's a watery consistency. You kind of get the idea. We discussed it a little bit with those 3 different categories of toxins. So yeah, I don't think we needed to go in depth. Again, it would be a stool culture that we would grab, plate it out. We're screening for those pathogens that are listed under those slides. All right, so if you guys have any questions, as always, please feel free to reach out. I hope you have a fabulous week otherwise. Thanks.